Thank you very much. Thank you everyone for being here. I'm going to talk to you about my presentation entitled, working in gene regulatory networks in tomato. As an introduction, I want to mention you that plants as sessile organisms must adapt to several environmental conditions. And when any of these conditions switch to an unfavorable level, the plants must develop a response to stress in most cases. Now, these responses start at the genomic level, where signaling molecules are able to detect these shifts in environmental conditions and therefore trigger a signal transduction pathway. This pathway often is regulated by the effect of transcription factors, molecules that are able to activate and repress, depending on the response, signal regulatory cascades. Now, in most cases, the effect of transcription factors can be studied through gene networks, where we can represent a hierarchy of transcription factors that are able to regulate a specific physiological response or metabolic response, such as in this example that regulates nitrogen signaling response. Now, in most cases, there are two main types of gene networks: gene co-expression networks, where they study gene-to-gene interaction based on shared expression, and gene regulatory networks, where they study the effect of a transcription factor directly on their target genes. But for the creation of gene regulatory networks, we use omic information like transcriptomics, transcription binding experiments such as ChIP-seq, and also we can use experiments of chromatin accessibility. But now, a more interesting approach is to integrate multiple omics information to combine and increase the predictive power of gene regulatory networks. Such approaches have been done for Drosophila melanogaster, some plants like Arabidopsis and wheat, and other species that have demonstrated that a combined power of omic information in gene regulatory networks improve precision and accuracy in the detection of regulatory cascades. Now, for the case of my organism of importance, Solanum lycopersicum, or tomato, is a model plant due to its agricultural importance, standardized breeding, and self-pollination, and is widely being used for fruit development, studies in fruit development and pathogen interactions. But in the case, despite this, in network studies are still lacking in tomato. Most of the studies are focused in protein-protein interaction networks and limited to few gene co-expression networks with limited sets of data. Therefore, tomato networks currently lack accurate prediction of regulatory interactions between transcription factors and their target genes. Therefore, that's why we're planning this investigation to create tomato reference gene regulatory networks with organ specificities. Now, to achieve this, we first of all created a new annotation for tomato. If you have worked with non-model organisms, you may think that in most cases, different gene versions, different gene annotations, and different sets of experiments are variant between all the experiments available. So therefore, we try to combine this using the latest genome LL 4.0 and combine the annotations ITAC 4.0 and ITAC 4.1 into a new annotation called ITAC 4.1c. Also, we discovered a new set of transcription factors by manually curating all the list of transcription factors existing for tomato in this different annotation and in all the other websites. And also we included protein predictions for trying to understand which is a transcription factor. At the end, we ended up with transcription factors with more than 3 lines of evidence to be considered a transcription factor in our new list. Finally, we enriched the functional annotation for gene ontology for tomato by incorporating multiple sources of information. If we work on plants, you can see that some datasets like Plaza 5 can include more information about gene ontology information. Now, after this update of the annotations, we now compile and reanalyze all tomato omic information available in terms of transcriptomes, chromatin accessibility, and ChIP-seq. We ended up with more than 500 experiments of RNA-seq that was compiled and like 10,000 libraries of RNA-seq. All the chromatin accessibility experiments that were about 100 experiments, and all the available ChIP-seq libraries is available for tomato. With this dataset, we created a gold standard network for validating the gene regulatory networks we will build afterwards. Then, with all the other information, we produce with gene tree algorithm a new gene regulatory networks for tomato. We produced gene co-expression networks, and we created 2 transcription binding networks using promoter sequences and open chromatin sequences. And at the end, all this was integrated into the new reference regulatory networks for tomato. Importantly, all datasets were separated by the organ of origin, resulting in organ-specific regulatory networks. Now, studying the transcriptomes of tomato, we found an interesting result, that the majority of genes that were expressed between the different organs were shared between the different samples. The idea is that the majority of genes were ubiquitously expressed, and also we also find some genes that were expressed uniquely in each organ. And the same trend was observed for transcription factors. Therefore, the majority of expressed genes in tomato and for transcription factors are ubiquitously expressed across all organs. Now, as I mentioned before, we created our networks using GD3 algorithm that represents direct regulation, gene co-expression networks, promoting binding networks, and open chromatin sites networks. As we can see in these plots, the majority of edges, more than 50% of the edges of our networks were counted with additional validation evidence with one of the networks I mentioned just before. Particularly, we can see here that the majority of the importance or the contribution of other networks are for promoting binding networks, for gene co-expression networks, and in less level for open chromatin sites. Another interesting result that we found is that despite the contrasting result that I mentioned before, that genes and transcription factors are mostly shared between different organs, if we compare the pairings of transcription factor and targets as a whole, we now see that the majority of this information is unique for each organ in the plant, therefore suggesting a tissue-specific pattern of regulation between targets and transcription factors. This leads us to understand and trying to look how a transcription factor might regulate its targets. If we plotted here the transcription factor mean connectivity against the percentage of allegiance of a transcription factor, which means the percentage of targets for each transcription factor that could be shared between different organs, we see a direct relationship with the transcription factor with high connectivity also tends to have shared number of targets of target genes shared between different organs. Actually, this represented the majority of transcription factors that regulate cellular important functions. As conclusion of these plots, I have to remind you that tomato regulatory interactions are highly tissue-specific, and we find that highly connected transcription factors have the more conserved target genes across all organs. Now, to validate finally our networks, we decided to make an emphasis on a biologically relevant cascade for tomato, that in this case is fruit ripening response. We selected two transcription factors, RAN and TGL1, and tried to look for the targets of these two transcription factors in our networks versus experiments where they study the regulatory targets, such as KO experiments, and their binding targets, such as ChIP-seq experiments. Here we found that more than 70% of the targets that were found in our network were also binding regulatory targets found in these experiments. And for ChIP-seq, more than 50% of the targets as binding targets directly for other experiments were also found in our networks, and more than 80% for TGL1 transcription factor. This same information is represented in these boxes, where each of the blocks represent an experiment where they have the amount of genes that were differentially expressed or obtained as binding targets or regulatory targets of these TFs. And on the last line, we can see my fruit regulatory networks, targets for these TFs, and we can find that the majority of each were significantly enriched. Now, if we focus in fruit ripening responsive genes, the main cascade that we are studying here for these two transcription factors, we can see that these networks represent the targets found in my networks, in the reference gene regulatory network, and the colored edges represent targets that were also found as binding targets in ChIP-seq experiments, in blue for TGL1 and in pink for RAN. This result helps us to understand that gene regulatory networks gather crucial fruit ripening responsive genes validated for RAN and TGL1 transcription factors. Now, another interesting result that we can obtain from networks is that we can analyze it to obtain important hubs and new transcription factors that could be controlling a significantly biologically relevant cascade. If we study the fruit ripening responsive genes, we now obtain two new transcription factors, an ERF and an ARF transcription factor that were previously connected to other functional responses in tomato. This was involved in cell stress, this was involved in axillary shock development, and now these results point out that they could be important regulators of fruit ripening response and could be strictly correlated with other key responsive transcription factors in fruit ripening response. Therefore, the network analysis of our reference gene regulatory networks aid in the detection in the new potential key regulators in regulatory cascades in tomato. To finalize my talk, I want to mention that all my tomato organ-specific regulatory networks will be available in Tomby's web app. Like it's an open source platform that will be gather updated process omic information from multiple crop species, including tomato, Arabidopsis, and multiple others. It's a collaboration that we're doing with Dr. Tomás Matos in Universidad de Valencia. And here we can see representation of our my networks, like the one I'm shown before, obtained here in the website that will be available soon. As final remarks of my research, I want to mention you that the GeneTree-generated networks, enhanced with additional omic information, were able to demonstrate a significant enrichment in vivo validated regulatory interactions, and that our tissue-specific reference regulatory networks offer a valuable framework for identifying novel central regulators in tomato development and environmental responses. At the end, I want to acknowledge all my team, my laboratory, our funding sources, and all to you, my collaborators, and all of you for being here. Thank you very much.
Questions? I got one. I was just wondering, not so much of a question so specific, but with the latest years, we have seen, and tomato is one of these crops that has got a pangenome, with a ton of cultivars, different varieties that are grown and sought after their traits. So I was wondering, how would you integrate, let's say, these pangenomic resources to your work on the networks?
This is a great question. Well, the power of GeneTree, the algorithm I use for creating the networks, is that the more differences, the more experiments, the more treatments you gave them, the more target, the more data sets you gave them, is extremely useful because it is not, biased into to a specific response, to a specific variety, or a specific treatment. Therefore, my network is able to, you can filter the network to extract the targets of the genes that are responsive in a specific trait, or a specific treatment, or a specific variety, and it can respond the same way. Because despite all the varieties and differences they have in tomato, they still use the same reference genome for mapping. And maybe, a genome was better, but will be better. But still, many of the response in terms of treatments can be responded with this kind of network. Hopefully in the future we can separate each of it and use a genome instead of a specific genome to obtain all these data sets and recreate these networks in this other pathway. But today, it could be extremely useful for responding this kind of responses.
Thank you, Jose. We have time for questions.
Yeah, sorry, it came late, so I'm not aware you said what last. I'm just wondering about replication, and so how many biological replicates did you use to create the network? And if you consider there is a minimum number of replicates to build trusted networks?
Well, thank you. Good. It's a very nice question. Well, we use more than 10,000 libraries for RGRNs for creating the network, or is for providing to the GeneTree algorithm. When we studied these libraries, we didn't study it as an experiment separately. We tried to merge all the libraries into one data set, a huge table of counts. The only filter that we have is that we have more than 1 million reads, and more than 80% of the reads were mapped exactly to the tomato genome. The other problems that we could have in maybe the lack of biological replicates, this information, this statistics that is important for a single experiment of RGRNs could be a little bit dismiss when you try to correlate or merge this kind of amount of data.